Great, we can go ahead and get started. Thank you everyone for joining us today. It has been so exciting to see how many folks, experts from our field and from adjacent fields, how excited everyone is to learn from our speaker today. This is a brand new series on the science of alt protein. Each month we will dive into a new topic and we'll have a scientist or researcher from our field come in and tell us about the latest advancements in their research and also help us identify underexplored questions that they believe other young scientists and engineers, others in the field should spend time digging a little bit more deeply into. So before I pass it off to Birgit, I will give a few quick announcements. The first is that last week, GFI announced our 2020 grantees, which is very exciting. Our new cohort, I believe it's 21 new grantees, and you can view them here. I've just dropped a link in the chat. And the second announcement is that we also launched a new research funding database, which you can see at gfi.org/funding database. And that is basically a resource that we've compiled. It is small but growing, and it highlights a number of research funding opportunities that we believe will be useful for those seeking funding for alternative protein research. So we hope that that will be valuable to you all. And the last thing I'll say is that Birgit is one of many experts on this call. We are all so excited. She has a fascinating talk prepared for us. But we're also hoping that this community serves as a forum for exchanging ideas and exploring questions that the group has, and we want it to be a very interactive forum. So please keep that in mind. Don't be shy, come off mute, show your face. We are all friends here. And the flow of this call, we started off with a few announcements. I'll pass it off to Birgit to give us her presentation, and then afterwards we'll have some time for Q&A. Once we're done asking Birgit any questions that we have regarding her research or her work more broadly, we will also be able to expand to a broader discussion that anybody who has questions about alternative protein in general for others in the group that they'd like to discuss can then raise them. And I sent a note before this seminar, but some of GFI's own scientists will also hang on for up to 30 minutes after this call for any questions that you might have. So without further ado, please help me welcome Dr. Birgit Dekkers. Thank you so much, Birgit, for joining us today.
Yes, well, I would like to thank you for the invitation and giving me the opportunity to start with the presentation. Now I have to, of course, go to the next slide and try to get most of the faces a bit on the side so I can see my own slides as well. Well, first I give you a short overview of the content of this presentation. I'll give a short introduction about myself, an introduction about the main topic of today that I would like to share with you, which is a new technology, which is called Shear Cell Technology to create fibrous structures for plant-based meat analogues. I'll show you some short results about not plants, but a dairy protein, calcium caseinate, where the whole research line basically started. Then I dive into structuring of plants and how to predict structuring potential of plant proteins. And I like to finish off with a bit about the startup company that I have together with my co-founder Ernst Bril, which is called Rival Foods, where we aim to commercialize this unique technology. So I have a background in Wageningen University and research, where I started off in 2009 with a Bachelor in Nutrition and Health, much focused on healthy foods, sensory science, food digestion and health, which I really enjoyed. But I felt the urge that I wanted to contribute myself to healthier food. And therefore I switched my studies and went into food technology. During my master's, I did a quite special program with a lot of international people, 14 different nationalities, and we traveled around Europe to different universities. We were in Wageningen, in UCC Cork, in AgroParisTech in France, and in Sweden, in Lund University, where we got insights and learned a lot about food technology. A big topic was sustainability as well, and this was very interesting. And I did a graduation project at the Nestlé Research Center in Lausanne in Switzerland. And this all prepared me for a lot of nice, prepared me in a lot of nice ways in food technology. But after my studies, I still felt the urge to dive deeper, and therefore decided to do a PhD project at Wageningen University in the food process engineering group, where Professor Adriaan van der Groot was working already for decades, a decade on a novel technology which is called shear cell technology. And I started in 2014 my research on this topic, and I defended my work in 2018. My thesis was called Creation of Fibrous Plant Protein Foods. And most of this presentation, I'll tell you a bit more about the research that we did over those 4 years, also where we are right now, and which developments we've made over the past couple of years. And after my PhD, I decided research was not enough, and I wanted to continue with this great technology. And I founded a company which is called Rival Foods, where we aim to commercialize products that are created with this technology and create real plant-based culinary creations. To start really at the beginning with this presentation, I have to go to the aim of the food structuring group. This is a department within Wageningen University where the general idea is to study food structuring processes to better understand the behavior of dense biopolymer mixtures under conditions like mixing, extrusion, these types of processes. Where you have combined mechanical effects, thermal effects, of course also separate only mechanical effects and thermal effects. And this scientific understanding of these types of processes was something that they were aiming for. However, often what was felt is that it's very difficult to model these kind of mechanical effects when there are many different types of flow patterns inside. As you might know, in a mixer, and even more so in an extruder, there are a lot of different types of flow patterns, and this makes it often a black box, and understanding is not trivial. And therefore modeling it is even more complicated. Nevertheless, this was the aim, to better understand and model these mechanical and thermal effects. And then the idea was that you can better design your extruder-like processes. Well, to do so, to better understand these behaviors of dense biopolymer mixtures under flow, you can think of how to simplify these things, these processes. For example, by using a regular rheometer. Because the shear stresses that you can apply in a rheometer are comparable to extrusion conditions. Although, of course, the flow field is more homogeneous and well-defined, and you have a plain shear field instead of all these complicated flows. But this was the approach that was taken in the very early days, I have to say. However, as soon as you put a more dense biopolymer system into a rheometer, you are challenged with high torque values or higher viscosities, such that you reach torques that are not able to measure with a conventional rheometer. Often what we also saw is that the gap is basically too small to load your sample, and therefore all the types of measurements that you try to do are difficult or even impossible. For interpretation, it's for sure not possible, but also even just simply measuring it in practice was difficult. And therefore, the idea came up to develop our own rheometers that were able to do these kind of things. So then a shear cell was designed in the group of Wageningen University, which was based on a rheometer concept, where you really have basically the idea of a stationary plate, a moving plate, where you can have simple shear flow deformation of more concentrated systems, concentrated biopolymer systems, such that you have these shear stresses comparable to extrusion conditions. And one of the crucial things here as well is that you could heat this whole system, because most of the processes in, for example, extrusion are of course combined shear and heat, and therefore also these systems should be heated. And then the first two designs came up, a conical design where you have a cone-in-cone device where material can be placed in between, and a couette device where you have two cylinders. Basically, in between these two cylinders, material is loaded. And then the inner cylinder is mostly rotated to deform products. And one of the very nice things of this design, similar to a rheometer, is that you can independently study shear rate, temperature, and residence time. And this is something that you cannot do in a normal extruder, where all these parameters are coupled. And of course, the torque measurement was something very important, and was measured can be measured over time and online. I already got the question, can you explain how this is different than extrusion? So one of the aspects which is different than an extruder with shear cell technology is that you have there always is the shear rate, the temperature, and the residence time are coupled. So as soon as you start to increase the shear rate, the residence time in your extruder typically becomes shorter. And also the temperature is affected if you increase the shear rate. Moreover, one of the things that you do, and something we in the design of a shear cell try to prevent, is the intensive mixing that you have in a barrel, where these complex flow patterns are mostly arising. For the sake of research, they try to simplify this, but this also has an impact on the structuring in the end, which I will tell you more about later in this presentation. And then, of course, in an extruder, you have a second phase, which is called, or is often referred to as the cooling die, where you have most of the texturization, where you get laminar flow profiles and plug flow, depending on the viscosity of your matrix. Also here, I'll tell you a bit more how this is different than what we do in a shear cell later on. I first wanted to start with structuring with shear cell technology with, I think, what is a nice story. Because basically the fact that we can create fibrous structures with shear cell technology is a discovery, which was not intended at all. We had students working in the laboratory, and accidentally one of the students used calcium caseinate instead of sodium caseinate. And this was just pure luck, you could say, or a stupid mistake, just how you see it, because by chance the student found these very nice and interesting fibrous structures. Another student passed by and looked at these structures and said, "Oh, that's very interesting. What did you do there?" And the student was a bit Oh, I actually made a mistake. I used the wrong protein source. And then only a day later, the supervisor of the student passed by and looked at the structures. And then actually a whole research line started. So this is always a funny story. So these are calcium caseinate structures where you see a very nice fibrous structure on a larger length scale, as you can see on the ruler on the bottom. Thick and thinner fibers that you can even pull apart very nicely. This was done, this study was further investigated with also adding a second phase, which is fat. So you had combinations of proteins and fat, and at different shear rates, different times, different concentrations. And this all led to very nice and interesting structures on different length scales. So on a macroscopic scale, you could of course see these nice fibers, but also if you start looking with, for example, a confocal scanning laser microscopy, you could see these nice anisotropy and these fibrous strands. Here you see in green the protein phase and in red the fat phase. And how you could structure basically with a shear cell these types of materials. If you zoom in even further, of course, with a different microscope, in this case, it's scanning electron microscopy, you could see that you have structure formation down to a very small length scale, 100 to 200 nanometers length scale. You see anisotropy of the proteins. And at that time, it was believed that this is crucial for these structure formation to have structuring on such, down to such a small length scale. This was all done on dairy protein, and of course, that was a very interesting and nice results. But moving towards a substitute for meat, this was not satisfactory in terms of sustainability. And the idea was to see whether this shear cell could also be used for structuring plant proteins. And we did with many different ingredients, with protein flours, protein concentrates, protein isolates from a wide variety of plants, soy, wheat, pea protein. At this time, we are also looking into even more, you could say, exotic types of proteins from waste streams, or side streams, I should say, from different types of plants. And it all depends basically on the ingredients, the formulation, and the process conditions, what type of structures you can get. Here I just show you a small overview of all types of structures that we've obtained. Homogeneous gels, layers, thin fibers, thicker fibers. And for the rest of this presentation, I am going to focus on how to create fibrous structures, and also what we believe what is a fibrous structure. This is a reflected light microscopy picture of a fibrous structure, where you see that if we pull apart the structure, you see these nice strands separating from each other. And I believe that this is also when we start eating and consuming these types of products, is what you feel in your mouth and giving you that meat-like mouthfeel. Before I dive into the content further, I would like to show you what an experiment on a shear cell looks like. In this case, I blend some plant proteins with water, and you obtain a dry dough-like mixture. This is actually what a shear cell in practice looks like. So 2 cones. The whole system in this case is already preheated, and the bottom cone starts rotating, the top cone remains stationary, and that's what the shear process looks like. Then upon cooling, you can open the whole system again and take out a sample. Often what we do is we fold these types of materials and look at the structure. And what you can see then is how the fibers start appearing. And this is a device of 100 grams where we do most of our early stage research on. 100 gram is enough to look at the types of structure. You can vary the time, the temperature, and the shear rate. And it's a great research device. One of the things that we then started doing is tensile tests. I hope I can also start this movie already. Basically taking samples in parallel and perpendicular to the shear flow, and then see how the material behaves upon stretching these types of systems. In the case you get a nice gel, the strength in both directions, in parallel and perpendicular, is the same. In the case of a nice fibrous meat-like texture, you see that the material strength is much higher parallel to the shear flow than perpendicular to the shear flow. And one of the biggest research lines where I've worked on is understanding how this is possible and why under certain conditions you can create these nice fibrous textures and under other conditions you cannot. And this is the hypothesis that we came up with that I would like to share with you, and hope to be challenged by with you later. One of the things that we found during our research is that you often use a two-phase system, which is phase separated on a microstructural scale. So if you blend two ingredients, it can be proteins, it can also be proteins and polysaccharides, for example, soy, or so in the case of soy protein concentrate, you have the proteins and the polysaccharides in there, and these two are phase separated on a microstructural scale. What we then do in the shear cell process, we are deforming and aligning these types of ingredients in the shear flow direction. Upon cooling, after deformation, the two phases solidify, and these two phases have a different strength. And that's the whole basis of creating fibrous structures. And I'll explain a bit more about this. First of all, let's start with this first thing. You have a two-phase system that is phase separated on a microstructural scale. We have analyzed many different microstructures. And one of the examples that we've seen a lot is a protein isolate. If you use a protein isolate alone, so a one-phase system, you see also in its microstructure no anisotropy. Then if we add a second component, a polysaccharide or another protein, you see a phase separated blend on a microstructural scale. Then if you bring this into the shear cell and you apply the right conditions, then you see that this second phase can be deformed and aligned in the shear flow direction, as you can see on the right side in the picture. So on the left side, you see a kind of blobs, a kind of spherical droplets inside of another matrix. And on the right side, you see that these blobs, these networks are deformed in the shear flow direction. In this case, these blobs can be even up to 300 micrometers, for example. So what does this do? This microstructure, what we hypothesized is that this dispersed phase that you see in the microstructure is weak, or that there is weak adhesion between the dispersed and the continuous phase. If you then start pooling these types of structures, in the case that you are not able to deform the structures, then you see that the weak surface is in both directions the same. Whereas if you are able through our process to deform and align these types of dispersed structures, and you see that the wake area is much smaller if you pull apart these structures in parallel to the shear flow direction than perpendicular to it. And the more you're able to deform and align these types of dispersed faces, the larger this difference becomes. And the more pronounced the fibers you get. So we call this a capillary filled gel, where you have anisotropy in your dispersed face. And what you see macroscopically, and when you deform these types of systems, the fibers are appearing when you deform the product along the long side of a weak dispersed face. And basically the fibers that you see are the matrix, the continuous matrix in between the two dispersed phase droplets that are aligned in the shear flow direction. And this is actually what we see and what when you start eating these types of products, really feel as the fibers similar to meat. Because in meat, you also have these types of fiber structures and this anisotropy in your matrix. So this is basically the hypothesis that we posed. And one of the key aspects that we then wanted to understand is what are now the properties of the ingredients that you need to be able to do this? Because as I told you before, we've studied many different types of ingredients, different types of process conditions. And in some cases, you do get a nice fiber structure. And in another case, you get a gel-like structure or layer-like structure. So the aim of the research then became how to predict whether you get this type of deformation and alignment during shear cell processing. So here's the question again: how to predict whether two faces in a blend are deformed and aligned in the shear flow direction. And then we can go to some physics, where you can see whether faces are deformable or not. In this case, you have basically two extremes, you could say: isostrain deformation and isostress deformation. Isostrain deformation is, for example, if you have a cheese with holes in it, when you start deforming the cheese, you're also certain that the holes, which are weaker, that they are deformed in the direction as well. On the other side of the spectrum, you have the isostress deformation. You can see this, for example, as water with stones inside. When you're able to deform the water, then that doesn't mean that you're able to deform the stones inside as well. So then what you do, you have a deformed matrix, but the dispersed phase is never deformed. Well, to create these types of fiber structures, you want to deform both phases, both ingredients, and therefore you want isostrain deformation. So the idea was to predict whether an ingredient under certain process conditions can create a fiber structure. You need to have a better understanding of the deformability of the two phases by determining the rheological properties. However, the rheological properties of these types of blends is quite complicated. That is because you have concentrated systems, concentrated biopolymers that have an interaction with each other. Although they are not mixing, they are phase separated. At the same time, they are still affecting each other by its behavior. And what is crucial to predict whether you can get isostrain deformation, that depends on the protein concentration that you have in each phase. And that again is dependent on the water distribution. Because of course, these types of systems, you have approximately 70% of water, between 40 and 70% of water typically. So to determine the rheological properties of the phase, you also need to understand how the water distribution is between these two proteins. And secondly, you need to understand the conditions under which you measure these types of properties. In shear cell technology, and this can also be translated, I guess, to extrusion, you are often dealing with these types of proteins and viscosities at elevated temperatures, between 100 and 160 degrees. And of course, the viscosity of the proteins and the ingredients that you have are dependent on these conditions. So to start off with the first challenge that we have, water distribution in phase-separated blends. Water distribution basically is affected by the properties of your ingredients, by the water affinity, the water holding or water binding of ingredients. This is different for different types of plant proteins. So soy has different water holding than, for example, wheat proteins or pea proteins. Also, the type of ingredient that you have, so if it's a protein isolate or a concentrate, affects how much water a phase or an ingredient will absorb. And then, of course, the extraction methods that are used strongly affect the water binding and holding of proteins. So this is all something that you have to take into account when you start to investigate these types of things. And then what you can do is investigate how much water is absorbed by these two ingredients. And we did this in different ways. We simply measured how much water is bound by the separate components, but we also wanted to see how the ingredients affect each other in water binding. And one of the things we did is just visually look at these types of systems when they are blended. In this case, we had a protein-protein system, so two different types of proteins, which resulted in really nice fibers. And we blended them in different ratios and then stained the proteins with rhodamin B and looked at it with confocal scanning laser microscopy. And what you see here is that the proteins are indeed phase separated on a microstructural scale. And you see different types of affinities of the proteins for the rhodamin B, and also differences in light reflectance on the bottom side of this microscopy picture. What you can see, for example, in the 50-50 dispersion where we mixed both proteins in equal amounts, is you see that the continuous matrix was one protein with a dispersed the other protein. And you could see that the volume fraction of the dispersed protein was smaller than the continuous matrix. Well, this was already one of the ideas that we visually saw, that in our systems, the proteins are not attracting equal amounts of water, but one protein attracted more water and was therefore in a more diluted form present in this mixed system. To further investigate this, we also used and looked into time domain NMR, nuclear magnetic resonance, to look at water binding. And here we also could clearly see differences in water binding between the two proteins. And we found out that it's true that what we hypothesized, that in a two-phase protein system, water is not distributing evenly, and that the mass fraction is not directly the volume fraction of a protein. And this was then used to actually determine the rheological properties later on. And that's then the second point that I wanted to show you a bit more about today, is how to measure rheological properties and how to use rheology as a tool to predict structure formation in shear cell technology, but also maybe later on in extrusion. You can only measure these types of viscosities under relevant conditions, because measuring rheology at room temperature if in the end your process is at elevated temperatures, above 100 degrees, then the viscosity at room temperature doesn't really say you anything. Therefore, we're using a rubber process analyzer, which is maybe a bit an odd thing, because this rubber process analyzer is, as you can imagine, not developed for food, but for rubbers, elastomers, tires, these types of things. This was quite a big challenge, because the people that we were working usually with these types of devices didn't hear anything about food and never experienced working with food. But it was a very nice device for us to work with, because it could measure rheological properties above 100 degrees in a closed environment, in a closed cavity. And thereby we could measure these rheological properties in a nice and in a good way. And what we found then during this research is that if you determine the rheological properties of ingredients, that at water distribution that is even between two proteins, which we in the early days assumed, you see a very large difference in viscosity of these two types of ingredients. However, when we took into account the uneven water distribution as what we found during our research with the confocal scanning laser microscopy and the time domain NMR, that the rheological properties of these ingredients actually was much closer to each other, much more similar. And this is then something that we thought that is very interesting, that you need viscosities that are close to each other to be able to deform and align these types of proteins to create a fibrous structure, what we of course were interested in. Well, these are some of the findings that that we published over time. A combined NMR approach for determining water distribution in protein blends could be interesting, an interesting read, and a closely related article is the second one on phase properties of soy and wheat in a blend for fibrous structure formation. Other readings on how to understand the deformation of two phases and how this leads to fibrous structure can be found in the second and third article, and an overview of different types of structuring processes for fibrous plant protein products in the last article. So this is the group there where I did most of the research during my PhD. On the right side in the front, you see Professor Adriaan van der Groot, who is actually the inventor of shear cell technology and who started off this whole research line. And yeah, these are all PhD and postdoc researchers in that food process engineering group, where also a lot of other and more interesting research is happening at the moment. Then a bit more about what I'm doing right now. I think the dream of every scientist is to see your research implemented in real life. And this is also something which I really wanted to do. Contributing to innovative, sustainable ways to meet world's protein demands is something that was also one of the reasons why I started researching shear cell technology. And now my life goal is to transform this interesting technology from a research tool where it was initially towards a more production tool to create culinary, sustainable and healthy plant-based meat analogues. So these are some pictures of the devices that we have. On the left-hand side, you see a conical shear cell. shear cell that we use in the lab to do most of the studies on new types of plant proteins, process conditions. And this device has been translated already to a pilot device, a scaling factor of 70 times, which is called a Couette shear cell, consisting basically of 2 cylinders. In between the proteins are packed for structure formation. Also here I have a short movie to show you what it looks like. Similarly, we are mixing plant proteins with water, and what you get then is a hydrated plant protein dough. We put this into a filling device to load the proteins in between the 2 cylinders. The process itself, the inner cylinder is rotating. Everything is heated at the same time and then cool down before we can open the system and take out our sample. What you can see here is 7 kilograms of material rolling off the machine. And with this technology, I believe that we can really offer a new type of product. Sorry. A new type of product in the meat analog space, where we looked at products that are currently out there. Very nice products, but often processed and productized. You see a lot of burgers, sausages, minced types of meat. And if you then compare this to the offer that you see in the animal-derived product space, where you have these nice whole cut products, there's clearly something still missing in the plant-based space, whole cuts. And this is something that we can uniquely create with shear cell technology. So what we aim to do at Rival Foods is to create plant-based products that are really addressing a culinary need from food professionals and chefs by delivering them the first whole cuts. Our products are large in size, as you could see in the movie, have a unique thickness of 3 centimeters, a unique size. Now the proof of principle that we have is 7 kilograms. And we can create different types of textures. Depending on ingredients, process conditions, and formulation, we can create layered structures, very soft, which we call Rival at Sea, a product which could mimic fish. Then Rival on Land, a thicker and tougher fibrous product, and Rival with Wings, a finer fibrous product. These are all still product prototypes, but we aim to develop these 3 product categories to replace products such as fish, red meat, and poultry. Our products are very high in juiciness. That's because we typically use 70% of water and can be either neutral in taste or already flavored and colored. The big difference between the structures that we can create with shear cell technology as compared to extrusion are summarized here. So with extrusion, you get in a cooling die a kind of V-type pattern that then can be pulled apart into, for example, chicken pieces, whereas we have this fibrous structure throughout the matrix, as you can see here, with a unique thickness of right now 3 centimeters. And this can be used in many different ways. Here is just some overview of work that has been done by chefs that have used their products and went from a large slab to nice fillets, loins. And of course, we are working towards the holy grail in meat, creating a plant-based steak type product. I was asked already also a couple of times, how does your process compare to extrusion in types of sustainability, in types of mechanical energy, for example? And this is not really my field of expertise, so therefore I cited one of my old colleagues who designed basically this upscaled shear cell, Couette cell. And what he has measured is that when you use the optimum process conditions, then the average specific mechanical energy is between 18.5 kilojoules and 32.6 kilojoules per kilogram. And this is much lower if you compare it, for example, to extrusion, which varies between 200 and 1200 kilojoule per kilogram. Of course, I am also aware that this specific mechanical energy is something which is different in both technologies. At the same time, the sustainability of meat analogues as a whole is much more dependent on the ingredients that you use than on the processing conditions that you use. But we do have slightly milder conditions than you see in extrusion. Well, this is the Rival Foods team at the moment. We are focused on developing the technology and the products itself on a small scale, still really taking it out of the lab into industrial application. And with this team, we are very happy that we are one of the few that receives GFI research grants to continue our work on this great technology. So yeah, we are very happy about this. And with that, I come to the end of my presentation. Thank you very much for your attention, and I hope it has been interesting. And do you have any questions?
Thank you, Birgit. This was such a fascinating talk. I think I speak for everyone when I say that I learned a lot, and I think we all did. I've been keeping track of a ton of questions actually that came through via the chat during your call. So maybe I will run through these, and people should feel free to hop off mute and contribute additional questions if they'd like to ask our speaker here some questions. So the first one is from Karsten, and she asks, how do you cool it and how long does it take to cool down?
Oh, that's a good question, and not an easy question, I have to say. It really depends on the starting point and the device that you use, how long it takes. So in the conical shear cell, cooling goes quite rapidly. This is because we are heating, as you can see on this picture as well, maybe through oil baths or water baths in some cases. So then the oil and water flows through those cones. It's a double wall system. And then you can cool down quite rapidly within 5 minutes. That's also because the thickness of such a slab is just 5 millimeters. Of course, the thicker the product becomes, so the 3 centimeter slab, this takes much longer, and it has to do with heat transfer. So if you're not shearing anymore and you have to cool or heat through such a thick slab, this takes more time, varying, yeah, roughly 20 minutes, I would say.
Awesome, thank you. The next question is from Lucilia, and they ask, what is the highest temperature the ingredients are exposed to during shear cell process, and does it affect the properties of the proteins? They're thinking specifically about the conformation of proteins and allergy epitopes in this case.
Ooh, that's a good question as well. So the processing conditions really depend on the ingredients that you use. We have so far experience with formulations, ingredients where we use maximum 140 degrees. And then the question about how it affects proteins. Well, basically what you do by heating the proteins is making everything more deformable. You get a kind of protein melt, and that's necessary to deform and align them. How this really affects proteins on a epitope level, I guess this is then moving towards allergenicity. I have to say that's not really something that I've studied myself. What has been studied within the university is digestibility, and we do see when having different temperatures that you also affect the digestibility. But this is not always negatively affected. It can also be positively affected by using higher temperatures. I don't know if this is the right answer that you were looking for, but...
Thanks, Birgit. The next question is from Aletta. She's wondering, is it possible to manipulate the phases of shear parameters to generate more loosely packed fibers?
Ooh, can you repeat that maybe once?
Yes. Is it possible to manipulate the phases or shear parameters to generate more loosely packed fibers?
Yes. So yeah, you can do this. It depends all, the answer is a bit the same as my previous answer, but it depends on the ingredients that you use, the formulation and the process conditions. And when you have an understanding of how these ingredients affect each other, then you can tweak it slightly towards, for example, more deformed phases where you get higher anisotropy. Yeah.
Awesome, thank you. And from Zach Weston, do you anticipate that shear cell can be done as a continuous process instead of a batch system?
Yeah, that's a good question that we haven't figured out yet, but it's something that we are thinking about as well, how to do this without it becoming an extruder, basically. Because of course, we went from an extruder to a shear cell to simplify and make it batch. The most logical way to make it continuous is going towards an extruder. However, then you affect, of course, the flow patterns, and you cannot independently steer these process conditions anymore. So it's something that we are thinking about as well, but I don't have the answer yet.
All right, next question is from Cam Davidson. In conical shear cell, approximately what temperature is the protein heated to, and approximately how fast does the plate rotate? Very specific question.
So the temperatures are between 90 and 140 degrees. But this is something we can change by changing the water or oil that is floating through the systems. And shear rates vary a lot depending on the ingredient type and formulation again. What is a good shear rate to apply? Our systems can handle between 0 RPM, so no rotation at all, of course, very slow, 5 rounds per minute. And we have experience with 150 rounds per minute, so very fast shear rates.
All right, we only have like 12 or 15 questions left. So
wow.
Bird, I wanted to clarify with you, are you able to stay on for a bit of extra time?
Yes, I am. I will put on some lights because I see it's getting dark here. I told you I'm in the Netherlands and it's...
Sounds good. Awesome. This next question is from Sarah Malferrari. She asks, what would be the ideal fiber diameter for plant-based meat, and how small can you go with your technology?
That's a very good question, and I have to say I don't know the answer. We are often thinking about this as well, down to what length scale do you have to go to really mimic the fibrousness of meat, and the same to also get the same chewing experience, flavor release, these types of things as meat. But to be fully honest, I don't know.
Hey, our next question is from Eli Gasgill. I was wondering how the shear generated by the Shear Cell Technology machine compares to the shear generated by a high shear mixture. This leads back to, mixture, but mixer. This leads back to Zach's question about continuous operation. Could an inline high shear mixer give the opportunity to manufacture continuously?
Well, a mixer, how can I see this? Basically, what we do is apply shear over a whole plate. Typically in mixers, you have smaller surfaces or at least connections to your material. So I'm not entirely sure how I would see this happening. More overly, usually with mixers, you have these types of complex flow patterns where the mixing near the rotating plate is higher than near the wall. And these, that gives you more complexity than the simple shear flow that we're using in a shear cell. But yeah.
Awesome. There's also a companion comment from Oscar, and he says, for continuous shear cell, consider a combination of couette flow with axial flow in the annular gap. Think of it as a variation of a single screw extruder instead of the screw you have, something that's more like a cylinder.
Yeah, that is interesting. It's something that we were thinking about as well, but still then you get, if you have the extruder like this and you have the rotation like this, you still have to move your material sideways, which gives you an additional flow direction. But yeah. Amy, somehow I cannot hear you anymore.
Oh, sorry. All right.
Now I can hear you again.
Thank you. This next question is about how whole cuts also seem to be the focus of several cultivated agriculture products. How would you shear cell process compare and contrast to this alternative method of production? So,
ooh, that's, well, one of the things is I think we are already closer to real implementation with plant protein foods than cultured meat is, but of course I'm not a cultured meat expert at all. As far as my knowledge goes, I know that it's quite difficult to give texture to cultured cells. And this is something that scientists are, I think, busily working on. I have no idea how far they are with this yet. And then lastly, there is, of course, the difference between plant proteins and cultured meat in terms of consumer acceptance. And I think, well, both products could move this field forward. So yeah, I would be interested to see what it looks like. And maybe we can even combine things, cultured meat and shear cell technology in the future.
yeah, that's a great answer. I think these two fields often like to view themselves as very separate, or these two parts of our sector, but in fact, in the future, they'll likely offer ingredients up for the end product. So Eli has a question. They wanted to ask how the protein powder would be mixed with water on a production scale, since the hand mixing method showed in the lab video wouldn't be feasible at production scale.
No, no, it's not even feasible for our 7-kilogram device. So then we use actually normal mixers that are also used in baking industry, for example.
Awesome. A question from Dana Hillel. Is the shear cell technology, does it damage the nutritional facts of the protein?
Ooh, again, this was not really a topic of my research line. I had a colleague, Patricia Estrada-Duc. She worked on the nutritional aspects of shear cell created structures and investigated digestion, but also enhancing these types of products with iron or vitamin B12. And I could forward her research outcomes and share it. I have to say that there are different outcomes that processing does affect the nutritional value, but no human trials have been done yet. And I do not suspect that the changes in proteins are very different for shear cell technology and extrusion, since the process conditions are quite similar.
Awesome, thank you. This is a related question that I think you answered a bit of already, but I'll let you comment if there are additional thoughts that pop up. Jida Rule asks, do these proteins denature at high temperature through shear processing? And if so, does this denaturization have any effect on nutrition?
Yeah, well, I have to say that we often used protein isolates and concentrates that were beforehand already denatured. So the denaturation of proteins is not something that is part of the process itself. And yeah, the nutritional value, I guess I already talked about. So
yeah, absolutely. Nadia asks, can this kind of technology be applied to the development of cheese substitutes? Have you ever tried to work with an emulsion like plant protein and a fat source?
So we've used fats in combination with proteins for the application of meat analogues. Not so much on cheese. This is because if you use very high concentrations of fat, you do get slip within our systems. Something similar you can see with normal rheometers if you use too much fat inside. The answer, yeah, the simple answer is no, we haven't, we don't have much experience with this.
Awesome. 7 or so more questions. You were doing great. We appreciate your fortitude. Mateus asks, how is the fiber diameter in comparison to meat? Which I also think you may have spoken to a bit already.
Yeah, so what I see as a fibrous structure in plant proteins is more like a capillary filled gel. So where you have a weak phase inside a protein matrix. This gives you, when you are tearing it apart, the same properties, but is not exactly the same as meat, where you really have fibrils that are built up, proteins, actin, myosin, building up this whole structure from a very small protein scale up to the thick fibers in your muscles. So this is something different, but I guess the anisotropy that we create in plant proteins does give you, in the end, the same mouthfeel. And I think that's key. Yeah.
Amazing. So Giuseppe from Novameat asks, do other ingredients other than soy and wheat work with your technology? He's thinking, or as his understanding, is that the material is subjected to a minimum of 120 Celsius degrees, which may not work well for pea, rice, hemp, and some other legumes.
We have experience with other proteins, not all of the proteins that you've mentioned. And it's true that depending on the plant protein, you use different process conditions. Yeah.
Great.
Let me quickly turn on the light because it now gets really dark. I'll be back in 2 seconds.
Oh, yes, sounds good. I'm sorry. I thought I had given you a chance beforehand, but I'm realizing I just kept bombarding you with questions. All right.
I guess this is better. You can see me now again.
Yes, this works well. Thank you. Another comment from Oscar. The difference between Ca and Na caseinates suggests some chemical cross-linking of the proteins can have a dramatic effect on the resulting fibers. Have you considered pre-treating plant proteins to achieve a similar effect and see if it yields better fibers? For example, perhaps you can treat plant proteins with some transglutaminase first and see what happens.
Yeah, it's correct. We haven't. There are some chemical changes that we've seen in calcium and sodium caseinate. We haven't worked with transglutaminase in plant proteins yet. I think for a scientific point of view, this would be very interesting. In practice, in plant proteins, it would become more expensive to do so. And therefore, we haven't tried it yet. But it is something that I think also within the research group, something that has been thought about and maybe already even conducted without my knowledge. Yeah.
Thanks, Birgit. You answered this question part ways already, but Poonam is wondering whether the technology holds true across all plant proteins.
Well, all plant proteins, I wouldn't say, or I wouldn't know. I have to say we haven't tried all, but we've tried multiple, and it's always a search for the right process conditions and formulations. But we have shown that it's possible with multiple plant proteins, yes.
Thank you. And Ruben is asking, how does this technology compare to cultivated meat in terms of sustainability? I think I can jump in here really quickly just to say that GFI is undergoing a life cycle analysis right now in-house using some real data from cultivated meat companies, and this will afford us in a few months probably the most accurate view as is presently available of the sustainability of cultivated meat. Birgit, you're absolutely welcome to comment if you have more to add, but I would look out for this publication that comes out in a few months.
I agree. Thank you for helping me out on this. Yeah.
Absolutely. Awesome. So Karsten asks, can you explain more about temperatures and digestibility? Karsten, that's a little bit vague. Birgit, feel free to take a stab at it if you have a clear answer, but Karsten, feel free to hop off mute and add a little bit more specificity if you're looking for a particular kind of answer.
He's quiet.
Yeah. Okay, maybe we will hop on to the next question then. From Harry Berto, is Rival Foods selling the device, or do you sell your own meat alternative?
Yeah, thank you for that question. We are now developing still the technology, so getting ready for production. And then the idea is that indeed we will sell our first products created with this technology. Yeah.
Awesome, exciting. Giuseppe asks if this technology is patented, and Robert Mitchell would like to know how much one of these units costs if you do plan on selling.
Well, the costs I cannot say anything about yet, unfortunately. That is because we are still in the development phase. The technology, part of it is published, and part of it is protected through patents, especially the application. Of course, what you have to keep in mind is that the basic of this device is a rheometer. It's something you cannot protect. But there are some smart tricks that you can do that are necessary for turning this device into something which is suitable for production, and that's something that has been protected, yes.
Awesome. Another question from Alexi is, what is the lowest protein concentration of a powder that you have managed to texturize?
Ooh, well, it's often combinations of ingredients that we've used. And then one ingredient contains more proteins than another. Typically, yeah, it varies between 50 and 80% proteins. So far, we only have limited results with using only flours.
All right. Ruben has asked, You've talked about the meat-like structure that is achieved with this process, but what about the taste of the final product? How does it compare to plant-based products that are already in the market?
Yeah. Well, of course, if you only use plant proteins, then the products taste like plant proteins. So what we've been working with is different types of flavor, spices, herbs as well to give taste to our products. And well, if I can quickly go back to this slide, they taste delicious, I have to say. And it's not only me who says that, also the food professionals and chefs that we've worked with.
If you need additional taste testers for your products, I'm sure this community would be overjoyed to fill that role.
Great.
Awesome. So Karsten has now followed up with a slightly more specific question. Does a high temperature during processing have a negative impact on digestibility?
I have to say that I don't know the latest results about this. We did see an influence of temperature on digestibility. But if I remember correctly, in some cases this was positive, and in other cases, other ingredients, this was the opposite, negative. But to yeah, you can reach out to me after this this presentation, and I can look up the right literature and send it to you and share it.
Awesome. Birgit, we have nearly made it to the end. This is the final question from a very, very engaged audience. You've obviously captivated us all. From Daniel, this may be a bit general, but they're curious. How well characterized is the input protein mix? Is it a general protein slurry, or are there specific amino acid profiles that provide particular results?
No, so far we have used normal protein isolates and concentrates that you can, that are commercially available for this process. So there's nothing special in that sense about it. If I answered then the question correctly.
Yes. I don't believe there are any other questions coming through. When you get the chance, maybe you can visit this chat, because it is absolutely bursting with enthusiasm. People are so excited about having been able to learn from you today, and we're all really excited about this product. Alexi actually snuck in a last question in there. Have you tried oat concentrate?
I don't think so, no. Not that I can remember, but that doesn't say everything about what has been done, of course, within the university in the broader research lines, but I don't remember that it has been used. No.
All right, awesome. Thank you so much, Birgit. Everyone is saying that they're so excited for a publication to come out of your newest research, and they're excited to follow the progress of your new endeavor at Rival Foods.